So today we will talk about fatty acid synthesis. You know what is fatty acid? So fatty acid is actually energy in our body when we take a sufficient amount of carbohydrate or more carbohydrate, then it will be stored in a form of fatty acid. Okay, so first fatty acid will be synthesized and fatty acid will be stored. In last class in lipid, I told you it is in adipose tissue, fatty acid is stored in the form of triglyceride. So first fatty acid will be synthesized, so stored in the form of lipid as high energy and least water requiring, because lipid is a very least water requiring. That is why it is stored. Otherwise, other molecule like glycogen, it requires water. It does not require water, so it does not contribute that much to body weight, but it stores pure. It occurs during well-fed condition. Obviously, the fatty acid synthesis, when you are well-fed, when you take a lot of carbohydrate, then only extra carbohydrate will be converted into fatty acid and lipid. And insulin activates the fatty acid synthesis. These are very important things. So insulin, ultimately what happens is, in the fed condition, insulin causes fatty acid synthesis. In other words, I keep telling that insulin makes you fat. Insulin makes you fat, and glucagon makes you thin. Insulin makes you fat. Okay, that is, you have to remember, makes you fat, because under influence of insulin, fatty acid synthesis happens. And where does this fatty acid synthesis happens? It happens in liver, kidney, brain, lungs, and mammary gland and adipose tissue. So actually, fatty acid synthesis happens in cytosol of almost all cells, and these are the tissue where predominant fatty acid synthesis happens. Liver is the very important tissue. Brain, because brain and nervous system, brain and nervous system has to synthesize myelin sheath. So brain and nervous system has to have a very high rate of fatty acid synthesis in order to maintain the membrane, in order to maintain that myelin. Kidney, lungs, mammary glands during milk production. Milk require fatty acid, so milk production during milk, adipose tissue, obviously, because adipose tissue has to store the fatty acid. So these are the main tissue where you will see more production of fatty acid will happen. Now let's talk about fatty acid synthesis as such. So you already know why does it happen. So actually I have answered in other words, why does it happen? So what is the role of fatty acid? Why? Where does it happen? So I have answered where question, and then so these are the why, where question, and you can see what regulates. So part of what question I have answered, what regulates, and then when does it happen? So you can see I have answered why question, where question, what, a part of what question, and when question. So I have answered this question through this slide. Now let's talk how does it happen, and along with how does it happen, we will talk about what are the regulatory steps, what are the enzymes, coenzymes related to regulatory steps. So we will completely answer the what question, and then how does this fatty acid synthesis happen. So whenever I always suggest you, whenever you study a pathway, just put this WH question: why, when, where does it happen, what, and then how. At least these 5 questions you have to ask. This five why question about the pathway. If you ask this 5 why question, then WH question, then your pathway, the most important part of this pathway, you will remember. So it will be covered. Now let's talk about how does this pathway. So fatty acid synthesis happens up to this palmitic acid. There is mechanism that is present inside cytosol. So fatty acid synthesis happens in cytosol, and this cytosol, okay, remember where does it happen? In cytosol of these all cells, hepatocyte, adipose tissue. For the time being, you will remember hepatocyte and adipose tissue, cytosol of hepatocyte and adipose tissue. So this cytosol will happen, and this happens up to 16 carbon. You know, 16 carbon saturated fatty acid is palmitic acid. So first saturated fatty acid will be synthesized always, and then after that, if our body requires longer chain fatty acid, then that 16 carbon fatty acid will be elongated in endoplasmic reticulum. In endoplasmic reticulum, smooth endoplasmic reticulum, not in cytosol. It will, this enzyme is present. The enzyme name is, simple name is the chain elongate enzyme system here in this endoplasmic reticulum. That will elongate this 16 carbon. You know, when you have to write next fatty acid, you will have to add 2 carbon, because 2 represent acetyl coenzyme. We will talk in detail about that. So further elongation will be done in endoplasmic reticulum. Now major fatty acid synthesis de novo is palmitate. We have already seen. So we will talk about the synthesis of palmitic acid. So when we talk about synthesis of palmitic acid, when you use that chain elongate enzyme, then further long chain fatty acid will be synthesized. Further addition of 2 chain will make it 18. Then addition of 2 chain here make it 20. So that is how chain elongation will happen, and that chain elongation happens in endoplasmic reticulum. Synthesis happens in cytosol, and chain elongation happens in endoplasmic reticulum. This is very important. Let's see this. What are the ingredients that are required? So here we will ask what question. All the what question we will answer here. So what are the components? What are the substrate? What is substrate? So what is substrate? And what is enzyme? And what are coenzymes? So we will answer this question here. You can see these are the substrate. You can see substrate plus coenzymes here. So you can see all the fatty acids are synthesized from acetyl coenzyme. Fatty acids are synthesized from acetyl coenzyme. What is acetyl coenzyme? This is acetyl coenzyme. This is, you have already know. You already know what is acid. This is acetyl, 2 carbon compound, and then this is a big molecule. Though I'm writing its coenzyme. This is a big molecule. This is nucleotide, a big molecule, much larger than this acetyl. So acetyl coenzyme A, from this acetyl coenzyme A, palmitic acid will be synthesized. So this form a unit. So 2 carbon unit, addition of multiple 2 carbon unit will form fatty acid. Now for synthesis of fatty acid, you require NADPH to join to form the bond between these 2 fatty acids. We will come to this point. And then we require ATP also for synthesis of fatty acid. For what? We will see that. So biotin and carbon dioxide. So this will be the part of this one reaction. This is done by one carboxylase enzyme. You know, whenever carboxylase is there, what does it mean? Carboxylase will the enzyme is followed by biotin. So biotin is integral part of this carboxylase, and then ATP. What does this carboxylase do? It will add carbon dioxide to form a longer molecule. So we will see this A, B, C. Here you can see this is a required acetyl coenzyme NADPH are required to elongate chain, to form chain. Now enzymes required, very important enzyme is acetyl coenzyme carboxylase. This is very important enzyme. This is rate limiting enzyme. Just remember this enzyme about fatty acid synthesis. If you remember this enzyme, so you will remember other things. This is the most important thing. So 80% of the question will be coming from this enzyme regulation and about the function of this enzyme. If you remember this enzyme, you will remember this is the carboxylase I told you here. So this enzyme will require this ATP, biotin, carbon. If you remember this enzyme, you will automatically remember this. Carbon dioxide will be added utilizing ATP and biotin. So you will automatically memorize this. And then if you remember the chain, you will remember acetyl coenzyme and NADPH. So these are the things you require. You can see in terms of vitamin, if you talk about vitamin, here coenzyme is equivalent to pantothenic acid. NADPH is equivalent to what? D3. So NADPH, always function of NADPH is, remember, reductive biosynthesis here. So what will happen? Fatty acid synthesis happens because of reduction. And that biosynthesis of fatty acid because of reduction is called reductive biosynthesis. Here, enzyme and second enzyme is fatty acyl coenzyme, fatty acyl synthase. So it is a big enzymes containing 7 enzymes. It is an enzyme complex containing 7 enzymes. One polypeptide having 7 enzymatic function. 7 different types of enzymatic functions. So let's talk about let's see this. So this acetyl coenzyme A, this is also acetyl. It will convert into 4 by addition of other acetyl. I am representing this acetyl coenzyme A by this line. You can just conceptualize like this. So this is coenzyme A. I am not writing here coenzyme because it will go away. So you just remember, I have represented this coenzyme A, acetyl, through this line. Remember this. Now this will form a bond like this. So between these two acetyl coenzyme, a bond will form, and this will result in 4 carbon. The same addition of next acetyl coenzyme formation bond will form 6 carbon compound fatty acid. Then again 8 carbon. Then the same way 10, then 12, then 14, 16. Then this is 16 is, what is 16? 16 carbon is palmitic acid. So this is palmitic acid. Did you get that?
Yes, sir.
Up to this. So this is actually, if you understand this, so this will be, I will elaborate this slide only. In whole of this synthesis of fatty acid, I will elaborate this slide only. So let's see this. I will first tell you about acetyl coenzyme. Let's see this acetyl coenzyme. What is this acetyl coenzyme? This is a protein. You don't have to remember that big protein. This is a multiple protein. So multimeric protein, acetyl coenzyme, it is a rate limiting enzyme, and it is a multimeric protein, you can see. And all rate limiting enzyme, as I have told you in the enzyme chapter only, they are allosteric enzyme. They are allosteric enzyme. Rate limiting enzymes are allosteric enzymes. So this is also an allosteric enzyme. And allosteric enzymes are generally multimeric enzymes. And it follow this type of curve, sigmoid shaped curve. You can see this. Now this is biotin. This is enzyme actually. This is biotin containing enzyme. What is this enzyme? Acetyl coenzyme A carboxylase. In short, you can write ACC. So acetyl coenzyme A carboxylase contain biotin. It will actually add COO group here. Here you can see COO group here, utilizing what? ATP. So that is why ABC. So carboxylation follow utilization ATP and biotin. So this carboxylation, this is so acetyl coenzyme will convert into malonyl coenzyme. This is acetyl coenzyme A. You can see this is acetyl coenzyme A, CH3, and this will convert into addition of here 1 carbon CO group. So CO2 group like this, COOH group. So this will form a malonyl coenzyme A. So what is malonyl? Malonyl, you can see a dicarboxylic acid. Malonyl is a malonic acid, is a dicarboxylic acid. You can see this is a dicarboxylic acid. It is a dicarboxylic acid with 1 CH through at the center. It is 3 carbon containing dicarboxylic malonic acid. So this acetyl coenzyme A will convert into malonyl coenzyme A. This is a very important reaction. It happens 7 times for synthesis of one for synthesis of one palmitate. Let's see this. And this second enzyme is 7 enzyme containing 1 polypeptide. This is called fatty acid synthase complex. This is a complex enzyme containing many enzymes together, but it is only one peptide. You can see many enzymes. We will talk about this enzyme. There are 2 enzymes that will cause reductase. So you can see reductase. So 2 enzymes. So this chain elongation will follow. So there is the bond formation between these 2 will follow a particular type of reaction. I call it CR/DR. What is this? RR means reduction. So 2 reductase you will find. C for condensation, D for dehydration. So you can see hydratase. Dehydratase you will see somewhere. Yes, condensation and then dehydration. So you can hydratase. You will see hydratase and then you will see synthase. And this is ketoacyl synthesis condensation. We will see about this all. Now, and these two are the initial reactions. So you can see the 7 enzyme complex, and there is one acyl carrier protein. So these are the protein where this acyl chain will be carried and elongate. Acyl chain elongate. So these are the proteins. So you don't have to remember about all this, though it seems complicated. It is not from exam point of view. This is not that important. Just remember, keep in your brain that this is fatty acid synthesis is done by a complex enzyme that one polypeptide has 7 enzymes, and those 7 enzymes will do everything. Along with this will be the enzyme. So this enzyme will help. Enzyme will start the reaction, and then further reaction will be done by this complicated dimer of this multimeric enzyme.
Sir, so what do you say CRDR stands for again?
Well, I'll tell you. Don't worry. I'll tell you. I'll elaborate in very detail. This is the enzyme. Okay. This is the enzyme. You can say this is enzyme. This enzyme. You can ACP. You can see two types of two. This is a cell carrier protein, and this part is synthase. So actually, there are two ends. I just want to show the mechanism. You don't have to worry about this. It is. You can say this. So you can see this acetyl coenzyme. This acetyl coenzyme will be added here first. This is okay. So acetyl coenzyme will be first added to this acyl carrier protein. Then now second, and you can see this has 2 hands. Though many enzymes, it has 2 hands. Just remember this fatty acyl has 2 hands. So this acetyl coenzyme will be shifted here to other ketoacyl synthase complex. Now again, this malonyl coenzyme will bind this acyl carrier protein, this second step. So this is step 2, you can say this. Now in third step, this 2 molecule will condense by releasing this CO2 from this. So it's not. You can say this. So the acetyl group from here, it is transferred to this malonyl. Now CO2 will release, and this will form this type of compound. Okay. So this is beta hydroxybutyrate type of butyryl coenzyme. Butyryl type of compound will be formed, and this will be attached here. So now dehydration, I told you, the dehydration will happen. Yes, see this. Now this cycle which goes on, okay, elongation cycle will go on. So if you have understood, that is good. If you have not understood, you will understand. First, what will happen? The acetyl coenzyme A, that will form in, so you know, from glucose, what will happen? So glucose will convert into pyruvate, and this pyruvate will enter mitochondria. In mitochondria, this pyruvate will convert into acetyl coenzyme A. Now through this acetyl coenzyme A, it will combine with oxaloacetate. So it is a part of Krebs cycle. And citrate synthase, you know this will convert into citrate. And this citrate will come out of the mitochondria. And this citrate will, again here in the cytosol, will break down again into oxaloacetate and acetyl coenzyme. Oxaloacetate and acetyl coenzyme. And this acetyl coenzyme A will be utilized. will be utilized, it will convert into fatty acid. So this whole process, you can see the synthesis of this fatty acid is happening from glucose. Okay, synthesis of fatty acid is happening from glucose. You can see one glucose is giving rise to two acetyl coenzyme, and this two acetyl coenzyme is being incorporated into this fatty acid synthesis. And this pyruvate does not directly convert into acetyl coenzyme. It goes through this mitochondria, and when it goes through mitochondria through citrate, so this method of transfer of this citrate from mitochondria, it's called citrate shuttle. This is transferred through a membrane channel. This channel system is called citrate shuttle. Citrate shuttle. So citrate shuttle will provide citrate to for acetyl coenzyme for formation of fatty acid. Let's see this. You can say this citrate come from this citrate shuttle from mitochondria. Now from mitochondria, one glucose will provide two acetyl coenzyme, and this acetyl coenzyme is a 2-carbon compound. Now this 2-carbon compound, after reaction with this carbon dioxide, addition of carbon dioxide, you can see one acetyl coenzyme comes here, and next addition to this 2-carbon will be added from this malonyl. Malonyl will come and add to this 2-carbon compound acetyl coenzyme. First acetyl coenzyme A will be there, but second the 2-carbon elongation will be because of malonyl coenzyme. This malonyl coenzyme A comes from this first reaction that I told you, the reaction of acetyl coenzyme A carboxylase that converts acetyl coenzyme into malonyl. Now this malonyl coenzyme A will come here and it will bind, and as a result only malonyl is 3-carbon compound, but from this 3-carbon compound only 2-carbon compound is added and CO2 will be released. Next step also you will see the same thing. Malonyl coenzyme A will come, and then same thing, the 2-carbon here added from this malonyl coenzyme. Again you will see 2-carbon is being added from this malonyl coenzyme. Where does this malonyl coenzyme come? From this rate limiting step. From this rate limiting step. So again the same thing happens. So this is a cycle of reaction. So same thing, same reaction, you can see the same reaction is happening 7 times. Same reaction is happening 7 times. 1, 2, 3, 4, 5, 6, and then 7 times. And this same reaction that happens 7 times will convert this 2-carbon into 16-carbon compound. Did you get that? This is the most important pathway, slide of pathway, you can see. So first a 2-carbon comes from acetyl coenzyme, but next 2 carbons comes from malonyl coenzyme. First 2 carbon comes from acetyl coenzyme, next 2 carbon comes from malonyl coenzyme. Here also, here also, here also you can see malonyl coenzyme, malonyl. And this malonyl coenzyme comes from this carboxylation reaction of this acetyl coenzyme. And in the beginning only, from the beginning only, I am telling you, giving you idea about carboxylation reaction. See, this is the importance. If you understand one reaction, carboxylation reaction, you will understand that anapleuritic reaction, pyruvate carboxylase reaction of Krebs cycle. You will understand the main rate limiting step of this fatty acid synthesis, which is the most, actually 80% question will come from this step only. And this step you can see, though only one step, only one reaction is there, it is happening 7 times here. Now this, after addition of this bond formation between this 2 carbon, 2 plus 2 carbon will be done by this 2 NADH every place. So how many NADH is required here? If you count for synthesis of one palmitic acid, 14 NADH. Because 7 steps are required for synthesis of one palmitic acid, and each step there is requirement of 2 NADH. 2, 3, 4, 5, 6, 7 steps are required. For each step there is 2 NADH. That is why this is, there is total how many NADH? 14 NADH. And this you can see, this is a rate limiting step, the whole rate limiting step that is happening 7 times. So reaction only. So first acetyl coenzyme A will be contributed. Other contributes first 2 carbon, and then next 2 carbon will be contributed by malonyl coenzyme. In 16 carbon fatty acid, the 14 carbons will be contributed by malonyl coenzyme A, and first 2 carbons will be contributed by acetyl coenzyme A. Remember this, very important. So now you can see this. So you can see the reaction by having 7 times. You can condense the whole process of difficult process into this 1 step. So what will happen? This 7 times process will happen. This same reaction will happen 7 times by utilizing this, and this will result in palmitic. The same reaction will happen 7 times. So if you remember this reaction, so you don't have to remember. Even if you don't, I told you that enzyme transfer, those all things, if you don't remember, if you remember that elongation of that chain, that is sufficient. That is sufficient. I'll tell you so very easily. So see this. This is one acetyl coenzyme A, and then this bond formation. Same type of reaction is bond formation. 2 acetyl coenzyme A come together, and then this 1 acetyl coenzyme A, this will be contributed by malonyl coenzyme. Second, this will first will be acetyl, second will be malonyl, and bond formation require 2 NADH here. Again, second malonyl coenzyme will contribute, and after addition of malonyl, 1 CO2 will be released. Remember that. So you can see formation of bond. Formation of bond again will be helped by this 2 NADH. Again same reaction. 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 So you can see this 7 times the same reaction will happen. What is this same reaction? This is, I called you CRDR reaction. Did you get that now? What is CRDR reaction? This is CRDR reaction. 7 times this CRDR reaction happens. Now, what is this 2 armies? Armies reduction. And that is why at one reduction, 1 NADH is required. At this reduction, NADPH, not NADH, NADPH. Remember, NADPH is required at 2 reduction step. Here you can see D and D. So condensation. C for condensation, because first reaction, this 2 molecule has to be condensed. So condensation, then reduction will happen. This requires 1 NADH. Then again dehydration will happen. Removal of H2O will happen. Dehydration. So it is done by hydratase enzyme. As I have shown you in that enzyme. If you go and see that enzyme here, CRDR. So you can see 2 reductase here. Enoyl reductase and ketoacyl reductase here. And if you can see DR. So C is condensation, keto-acyl synthase. This is condensation reaction. So it is done by this, condensation is done by this enzyme. Now D for dehydratase, hydratase. It is done by, it will remove water. Did you see this? Did you understand? The repeat of it is actually very easy. If you remember, though I have explained very much, that might have complicated the things, but it is very easy. This is simple reaction. So 2 acetyl, 1 acetyl plus malonyl coenzyme will come, CO2 will release, and then bond formation. This reaction, CRDR, 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 CRDR. Each step after addition of 1 acetyl, there is malonyl. Malonyl will come and CO2 will release. Malonyl comes, CO2 will release. Malonyl comes, CO2 will release. So this will form through 7 steps. It will form 16 carbon palmitic acid. Now, did you all understand?
Yes, sir.
Now let me now summarize all this fatty acid synthesis process. It's easy to now. Let me summarize, then you will understand everything. I have put the whole context in one slide. You can see from glucose, as I have discussed you, pyruvate will be synthesized, and this whole process is happening inside. This is a part of this the cytosol. Everything that I've shown here is cytosol. Now this pyruvate will go there, and this pyruvate through citrate synthase, and this oxaloacetate will join this through this citrate synthase, oxaloacetate plus this acetyl coenzyme A. Acetyl coenzyme A. So this will form citrate, and citrate will come through this transporter. This is called this transport. This system is called citrate shuttle. So through transporter, citrate will come, and citrate will break into oxaloacetate and acetyl coenzyme A. This acetyl coenzyme A will form malonyl coenzyme A, and this malonyl coenzyme A will convert polymerize to form fatty acid, fatty acyl coenzyme. And this fatty acyl coenzyme A, the last reaction is removal of this coenzyme. So this is called thiosterase reaction. Last reaction is thiosterase reaction, removal of this. Now this this is how palmitic acid will be synthesized. So you can see the process. So this, you can see acetyl coenzyme carboxylase. I have told you 7 steps. It is required at 7 steps. Then this repeated reaction is done by fatty acyl synthase and require NADH, 2 NADH per bond. And then last reaction is IST. So this is summary of fatty acid synthesis. So fatty acid synthesis, though if you study in a book and everywhere, it seems very complicated. But it is, remember, if you polymerize things, it will not be easy. Just if you remember that this fatty acid is a polymer of acetyl coenzyme, it is like snake. So it will not be that difficult. And this polymer, all polymer will form because of formation of bond. And here bond formation happens through CRDR reaction. CRDR. And then ultimately this fatty acid. So it is easy, very easy if you remember these steps. Transport of acetyl coenzyme I have already explained you. So acetyl coenzyme is produced in mitochondria, you already know in my matrix, by pyruvate dehydrogenase complex, and fatty acid synthesis occurs in cytosol. And that is why, so from this acetyl coenzyme has to be transported from mitochondria to cytosol. And this happens through this pathway. So citrate is being synthesized in mitochondria. Citrate will come out of the mitochondria, and the citrate will release acetyl coenzyme. This will be utilized for fatty acid synthesis. Now, this release of oxaloacetate will enter again into this mitochondria. So this is the pathway. You don't have to just remember citrate shuttle system. Citrate shuttle system, you can remember this if you want. There is malic enzyme, very important enzyme, malic enzyme. But if you don't remember this whole process, just remember citrate shuttle system provide acetyl coenzyme A required for synthesis of fatty acid. Okay, that is what you have to remember right now. Did you remember this? Citrate shuttle will provide the acetyl coenzyme A for fatty acid synthesis. Yes?
Yes.
Yes, sir.
Okay. Now, so this is summary. How this you can see the summary. So 7 acetyl coenzyme A, 7 CO2 and 7 ATP. You know this 1 acetyl coenzyme A plus 1 ATP will produce 1 malonyl. So there is 7 steps, that is a 7 acetyl coenzyme, 7 CO2 and 7 ATP will form 7 malonyl coenzyme. And you know ADP will be released because of a phosphate is consumed. 1 high energy phosphate is consumed. So ADP plus phosphate. So this results in 7 malonyl coenzyme A. And this is catalyzed by ACC, acetyl coenzyme carboxylase. You can see here. And this 7 malonyl coenzyme will be required in the reaction here. You can see 7 malonyl plus 1 acetyl coenzyme A will ultimately be converted into palmitate. This 2 will contribute carbon. And from this malonyl, 7 CO2 will be released. And for formation of this is 7-step process, that is why for each step require 2 NADH. So 7 step will require total 14 NADH. So you can see that this balanced reaction. So 40 NADH and not NADH. If I am wrong, you can understand NADPH. There is difference between NADH and NADPH. You will see, I'll tell you a big difference. NADH from catabolic reaction. From catabolic reaction, glucose if converted into pyruvate, it will be synthesized. But this is catabolic reaction. So NADH release is related to catabolism. But NADPH is utilized for anabolism. From acetyl coenzyme A to fatty acid. It is anabolism. So you can see NADPH is required in anabolic reaction. And NADH is released from this glucose and it is converted into ATP. It is not utilized for synthesis of molecule. And that is the very basic difference between NADPH and NADH. NADPH is utilized for synthesis of molecule, fatty acid. So that is why we call it, function of NADPH is reductive biosynthesis. Reductive biosynthesis. And very important function, predominant function of NADPH is reductive biosynthesis. So if you remember, whenever I told you what is the function of NADPH, just Remember this term, reductive biosynthesis, RB, reductive biosynthesis. So did you understood this? Did you understand this? Now let's say this 7 dehydration steps are there, because you know the CR and D is D and R. CR, DR is there, but you can see here only 6 H2O is written. Why is there 6 H2O? Because CR, D, R. So each 7 step is there, each step there will be release of H2O. Then why is there only 6 H2O? I've written here. Can anybody tell me? Look at this step. Look at this whole process. So can you see? Here will be utilization of 1 water, okay? Because this has to be removed. And this will remove coenzyme A. Did you get this? And that is why there will be 6 water only. Now let's talk about regulation. This is also very important, but easy. You know, when does this happen? Fatty acid synthesis happens when? During fed or fasting condition?
Fed condition.
Yes,
Well
well-fed. Yes, well-fed condition. So well-fed condition means which hormone?
Insulin.
Insulin. So insulin is activator or inhibitor?
Activator.
Oh, yes, activator. So activator of acetyl coenzyme carboxylase. How does it activate? Activate through dephosphorylation. It is activated through dephosphorylation. So this is an activator. Insulin is an activator of acetyl coenzyme carboxylase. So the one very important. Now, what and then second concept I told you, reactant stimulates. What is the reactant here? Acetyl coenzyme is reactant. Isn't it reactant?
Yes, sir.
Yes.
So it should activate. It activates. Okay. So what is the product here? Palmitic acid. Okay. Is it palmitic acid?
Yes.
Palmitic acid will inhibit this enzyme, and then other glucagon will inhibit this enzyme. Very easy. Isn't it easy? If you know the fundamental, the regulation becomes easy. Very easy. If you don't know the fundamental of this enzyme regulation is the most difficult thing. Now you can easily understand. So you can see this. So in place of acetyl, which represent acetyl coenzyme, citrate represent acetyl coenzyme. Because this acetyl coenzyme comes from citrate. So you can see insulin and this reactant will activate this, stimulate this ACC. And then glucagon and fatty acid, that is palmitic acid, inhibit. So how glucagon inhibit the enzyme, you know, through phosphorylation, dephosphorylation. Glucagon actually cause phosphorylation and it will inhibit acetyl coenzyme carboxylase. And this insulin will cause dephosphorylation, and that is why dephosphorylation will activate this acetyl coenzyme carboxylase. I'll tell you one thing. This, when you are in fed condition, what will happen is there is mitochondria. Here is, you can see this mitochondria. And glucose will convert into pyruvate, and this pyruvate will convert into, let's directly write citrate. Then citrate, when it comes in from this mitochondria into cytosol in high concentration, what will happen? pH will drop. There will be drop in pH. So this will cause what? This will cause polymerization of this enzyme. This will cause polymerization of acetyl coenzyme carboxylase. Many acetyl coenzyme will not form a covalent bond because non-covalent bond polymerization will happen. And this will form a large complex, and that is how the this acetyl coenzyme will be recruited and it will be converted very rapidly, and this all enzyme will be activated through polymerization. So this is a very different type of mechanism, very interesting type of feedback stimulation, where a substrate will cause polymerization. Feedback stimulation, feed forward stimulation cause polymerization. Citrate will cause FFS, feed forward stimulation, and because it is a reactant, that is why this reactant will cause polymerization. Now you can see this from this picture you can easily now understand. Glucose to citrate, acetyl coenzyme, malonyl coenzyme. This converts this acetyl coenzyme converts acetyl into malonyl, and you know this process already. So this is reactant, that is why reactant will stimulate. This is FFS. This is product will inhibit the FBI, feedback inhibition. Now insulin and glucagon will regulate through phosphorylation. This insulin will cause dephosphorylation, and that is why in active state. So this is related to well-fed condition. So this is how insulin regulates. This is how product and reactant will stimulate and inhibit the acetyl coenzyme carboxylase. So activated by citrate and polymerize this enzyme FF through feed forward stimulation, and you can see ultimate product is palmitic coenzyme inhibit the enzyme. So did you all understand regulation also?
Yes, sir.
So this is a very important. You can see enzyme regulation is you already know this. I don't have to repeat this. You already know. So you can see this. You can already know this. I don't have to explain all this. So, and phosphorylation, dephosphorylation is intermediate term. Long term, if a person keep on eating for a long term, what will happen? So insulin will be very high, and insulin will be very high, and this insulin will induce gene for transcription of mRNA. And mRNA will result in formation of acetyl coenzyme carboxylase, very high amount of acetyl coenzyme. This is long-term regulation, and this type of mechanism happens in obese people. So obese people keep on eating. Then what will happen? This high insulin will induce this acetyl coenzyme carboxylase, and because there is very high amount of this acetyl coenzyme carboxylase, it will cause more synthesis of fatty acid, and this is called induction. And opposite will happen during prolonged fasting condition, during intermittent fasting. That is why there is a new trend coming these days in fitness. So they suggest, actually, if you go to the gym also, they will tell you not to eat after 7 o'clock till the morning. At least you have 12 to 14 hours fasting every day, and this fasting actually induce that glucagon and induce this fatty acid catabolism of fatty acid, and that is how it will help your whole metabolism. So you require both. You cannot switch either side. You cannot be partial to one side. If you become partial to one side, you will be diseased. Okay? If you make your body to shift towards continuous fat condition, that is also bad. If you keep your body move completely shift towards prolonged fasting condition, that is also bad. So you have to maintain the balance. So this is why intermittent fasting has been very relevant. Don't eat after 7 o'clock. That is many fitness experts. If you ask, they will tell like that. Yes.
Can you repeat that, please? So why is it better to intermittent fast?
You can see this. If you keep on eating, what will happen?
Your insulin will increase.
And there will be a continuous high tide of insulin. So what will it do? Acetyl coenzyme. Did you get up to this? What will it cause? Fatty acid?
It will
fatty
cause
acid means fat.
acetyl coenzyme A.
And fatty acid means you'll become fat if you keep on eating. So what will be the opposite of this?
Glucagon?
Oh, yes, glucagon. So how will you get glucagon in your body?
Stop eating.
Stop eating. Yes, fasting. Fasting. Did you get this? So when you eat for 12 hours a day, and if you don't eat for 12 hours a day, that for 12 hours, what will happen? Fatty acid will be synthesized, protein will be synthesized. Your body is in anabolic phase. But when you don't eat for that 12 hours, from 7 p.m. to 7 a.m., what will happen? This process will reverse. Won't it reverse?
Yes.
Acetyl and then energy. So that will make a balance in your body. Did you get that?
Yes.
So that this is the actually a very important. Don't have those simple sugar. Have a complex type of sugar. Never have Coca-Cola. Coca-Cola is very toxic. I am very against that Coca-Cola. Any that soft drinks are very hard to body. Soft drinks are not soft to body. Avoid those sugar, simple sugars, and utilize this complex carbohydrate. And try to keep a gap between. Make intermittent fasting. Include your intermittent fasting in your body, in your daily regime. Have dinner before 7 o'clock and be in that intermittent fasting. Sleep early, and that these are the very, very simple things. If you use, you can make your. And I gave you an explanation for why intermittent fasting is important. Okay.
Okay.
Okay. So I gave you explanation through that, so that you will understand. Actually, you will understand the relevance of studying this fatty acid synthesis and fatty acid oxidation. Understanding the regulation. If you understand the regulation, then you will understand things. You will understand nutrition better. So you already know. I have told you this transcriptionally regulated. You go and read yourself. You will understand this. So insulin makes you fat. You know this. Did you now? Are you convinced now? Insulin makes you fat.
Yes.
So thank you. If you have convinced, my lecture is worth it. Okay.